So, very good morning to everyone. Myself, Dr. Prabhu, Assistant Professor of History of the Past. So, today we'll be studying about experimental proof of RNA as a genetic material. But what is RNA? RNA is a polymeric molecule which is essential for various biological roles. It is also termed as ribonucleic acids, and it is essential for coding, decoding, regulation, and gene expressions. So, several organisms are known to have RNA as a genetic material, such as viruses. They have genetic material, be it RNA virus having single RNA or double-stranded RNA. Several diseases are caused by RNA viruses. Say, for example, Rau sarcoma virus causes cancer, whereas HIV causes AIDS. SARS, influenza, hepatitis C virus also causes different types of DNA. So these are RNA viruses, sometimes retroviruses, which cause different types of deadly diseases. So here, if you see a retrovirus cycle, which enters a host cell through endocytosis, where the viral RNA gets converted by reverse transcriptase into DNA, which enters or integrates the host cell, again gets transcribed as a viral RNA assemble and as a replicated retroviruses, which further affects the host mechanism. So that's how it works. Now, this retroviruses works in two types of ways. Basically, one lytic cycle or lysogeny cycle. Now, if it enters lytic cycle, then phage attaches to the host cell, injects its DNA, where phage DNA circularizes and enters the lytic or lysogenic cycle. Now, if it is entering lytic cycle, this new phage DNA and proteins are synthesized and are assembled as a virions. And then cell lyses and releases this phage virions, which again gets move further and attacks the host cell. Whereas if it enters lysogeny cycle, it forms a prophage, where phage DNA integrates with the bacterial chromosome and forms a prophage. Now this reproduces normally, has several cycles of cell division, and then prophage may excise from bacterial chromosome by another recombination event, initiating a lytic cycle. So that's how these RNA viruses or retroviruses work. Next, if you talk of examples of plant RNA viruses, so tobacco mosaic virus was one of the example, which was crystallized by Stanley in 1935 for the first time. The TMV, which is tobacco mosaic virus, can also cause tobacco mosaic disease. It also causes disease in tomato, pepper, snapdragon, and marigold. There are several other types of plant RNA viruses, such as papaya ring spot virus or potato leaf rot virus. Next, if you talk of RNA viruses, here are the diagrammatic representation of different types of RNA viruses. So here if you see, the first is tobacco mosaic virus, which contains RNA nucleotides and protein. Next is HIV, that is human immunodeficiency virus. And then we have hepatitis viruses. Next, if you talk of discovery of RNA as viral genetic material, how it started. So all of the cellular organism has DNA as a genetic material. Some viruses, however, use RNA also. That's how we term it as RNA virus. So tobacco mosaic virus is composed of RNA and protein. As I told in the previous slide, it doesn't contain DNA. In 1956, Zider and Schramm showed that when purified RNA from tobacco mosaic virus is applied to the tobacco leaves, they develop this tobacco mosaic disease. Now, pre-treating this purified RNA with RNase, which again hydrolyzes the complete RNA, destroys its ability to cause the TMV lesions. So in 1957, Frankel, Conrad, and Singer showed that TMV infections with viruses containing RNA from one strain and protein from another strain. And the progeny virus were always of the type specified by the RNA and not by the protein. So that's how they concluded that it is RNA which is a viral genetic material, not the protein which is responsible for carrying of information to the next generation. So here are the images where MOP was used along with the TMV viruses. At last, you can see the mosaics can be developed. So what was Franklin, Conrad, and Singer's experiment? So here, if you see, there are two types of tobacco mosaic virus, that is tobacco mosaic virus A and tobacco mosaic virus B. So tobacco mosaic virus A and B both contains two parts, that is protein and RNA. So tobacco mosaic virus A contains protein A and RNA A, and tobacco mosaic virus B contains protein B and RNA B. So what he did was he just mixed and reconstructed in new types of virus by interchanging the protein content between tobacco mosaic virus A and tobacco mosaic virus B. So what he did, he reconstructed the tobacco mosaic virus by using RNA of tobacco mosaic virus A and protein of B, whereas protein of A and RNA of B. So here, if you see in both the cases, infection of tobacco leaf caused the tobacco mosaic disease, but then type A progeny, when it was checked after after it has caused the mosaic disease, it was found that it contains RNA A and protein B, and that is only responsible for further transfer of mosaic disease. So here is another diagrammatic representation which I told. So here, if you see, protein A and RNA A was reconstructed by RNA A and protein B, and virus had incorporated it, and mosaic disease was also occurred, also seen. So this proved that the specificity of virus proteins was determined by RNA alone, and the protein carried no genetic information. So therefore, it is RNA that is carrying a genetic information and not the protein. So the genetic material RNA is usually found to be single-stranded, but in some cases, say in reoviruses or wound tumor viruses, it might be sometimes double-stranded also. Now, experiment in 1960 showed that messenger RNA had the ability to store genetic information, and transfer and ribosomal RNAs have ability to translate these genetic information to proteins. So experiments performed several decades ago later showed that some RNAs can also act as an enzyme to self-edit their own genetic material. But then this created two major questions: that why does RNA play so many roles in flow of genetic information, and why bother storing genetic information in DNA if RNA is doing that job? So the result is the mutation rate. The mutation rate or RNA dependent RNA polymerase is substantially higher than that of DNA dependent DNA polymerase. And we don't have any double stranded DNA based organism other than viruses to look at. So this speculation is ruled out, and double stranded RNA is much less stable than double stranded DNA. So proteins usually fit in the major group because it is roomier, and the minor room is less accessible. That is why we are using most commonly as double-stranded DNA. Now, uracil adenine is also rather than weak and likes to wobble and bind to uracil guanine. So this base mismatches is also one reason. So that DNA is more commonly used as a genetic material. But then this was the experiment which give a proof that RNA is also used as a genetic material. Now, RNA has a greater capability as a genetic molecule, as it had to carry on hereditary process on its own. And it seems that RNA was the first molecule of heredity, and it is involved, including all the essential methods for storing and expressing genetic information before DNA come into the role. So single-stranded RNA is rather unstable and is easily damaged by enzymes. So by essentially doubling the existing RNA molecule or using deoxyribose sugar instead, DNA has evolved as a much more stable to store the genetic information with accuracy. Thank you.